Deep inside your tissues, an active rebellion is taking place, driven by cells that have forgotten how to die. This was first observed in the 1960s by biologist Leonard Hayflick. He discovered that normal human cells don't divide forever. They hit a hard biological boundary, stopping completely after about 50 population doublings. We now call this the Hayflick limit. It's triggered as the protective caps at the ends of our chromosomes, telomeres, shorten with every division. Once those caps are gone, or if the DNA is too damaged, the cell permanently locks its division cycle. This state of arrested growth is cellular senescence. The cell is still alive and metabolically active, but it has officially exited the cycle of replication. This sacrifice is a crucial defense. By shutting down damaged cells, the body prevents them from turning into malignant tumors. In this sense, aging begins as a strategy to survive cancer. In a young, healthy body, this isn't a problem. As cells become senescent, the immune system's macrophages and natural killer cells sweep in and remove them, making room for new functional cells. But as we age, our immune system undergoes its own decline, known as immunosenescence. This internal cleanup crew becomes less efficient, and its response time slows down. Without the immune system clearing them away, these arrested cells begin lingering deep within our tissues. They don't just take up space, they pile up, crowding out the healthy cells required for our organs to function. Because these cells refuse to die but no longer serve a function, they've earned a grim nickname: zombie cells. The very mechanism that protects us in our youth eventually becomes a localized toxic burden, driving the physical decline we associate with getting older. These zombie cells are not peacefully dormant. They are actively secreting a chemical cocktail into the body, effectively poisoning the environment around them. This diagram illustrates the source of toxicity. In response to damaged DNA, the cell expresses SASP genes. As the arrows indicate, it pumps out inflammatory cytokines and proteases, destructive agents that degrade healthy tissue and break down structural integrity. This creates a cellular contagion. The SASP secretions are so stressful, they can actually force neighboring healthy cells to shut down and become senescent themselves, spreading damage through the tissue. Aging, then, is a spreading process of inflammation that originates from these rogue zombie cells. When we look at the macro level, this microscopic spreading has a massive impact on the elderly body, compromising organ systems from the inside out. Scientists have directly correlated the buildup of these cells to conditions like joint degradation and osteoarthritis, and the lung scarring found in pulmonary fibrosis. Panel B shows the systemic result: chronic, body-wide inflammation. This constant background noise paralyzes the remaining healthy immune system, leaving it unable to clear pathogens or respond to new threats. This immune paralysis is why infections like influenza or COVID-19 are so lethal for older populations. The system is so busy fighting the internal fire of zombie cells that it cannot effectively combat an external virus. This suggests that diverse conditions, from lung scarring to increased infection risk, share a common driver: the persistence of these zombie cells. Rather than managing the symptoms of age-related disease, modern medicine is beginning to target the biological engine behind them. This has led to the discovery of Senolytics, a revolutionary class of drugs designed to selectively force zombie cells into the cellular death they've been evading. Researchers are currently testing combinations of existing drugs, like the cancer medication dasatinib and the natural plant compound quercetin, to achieve this targeted removal. Early results show that clearing these cells can halt the SASP secretions, allowing chronic inflammation to subside and tissues to potentially recover. By selectively destroying the cells that refuse to die, Senolytics offer a path toward reducing the inflammatory burden within the body. This research is now in the clinical phase, with early trials showing promise in treating diabetic kidney disease and lung fibrosis. However, the path forward isn't without complications. The body actually needs. Eradicating these cells entirely could disrupt those essential repair mechanisms. There is also the risk of off-target toxicity. Current Senolytics can accidentally damage healthy cells, such as those responsible for blood clotting. The future lies in precision. Scientists are now investigating immunotherapies like CAR T cells and specific antibodies to hunt zombie cells with high accuracy, bypassing healthy tissue entirely. While we may not stop the clock, clearing out these cellular ghosts means our years of good health can finally catch up to our lifespan.